Gentlemen and ladies, welcome back, and let's look at cardiac muscle now. We've looked at skeletal muscle and given the highlights. We are now looking at cardiac muscle. And remember, we start with the features. It is striated, meaning it has regular arrangement of sarcomeres. It is self-excitatory and electrically coupled. What it means is that you can remove the heart from the body and put it in a physiological medium or solution, and it will be able to continue with its rhythm of contracting and relaxing. Why? It has a conduction system, a number of cells within itself that are able to produce action potentials to stimulate the contractile cells. The rates of contractions, we said when we are looking at the different classifications, the rate of contractions is controlled by the autonomic nervous system. What are the cellular features? We find one or two nuclei or nucleuses per cardiac cell. And these cells are branched, connected one to the next through structures which are called intercalated discs. And in this structure, you find that there is intercellular communication. Communication by attachment, so there are adhesion molecules that exist. Also communication by sharing cytoplasm. That is through what are called gap junctions, the spaces between cells. Numerous mitochondria are found because this cell needs to produce a lot of energy for it to sustain the continuous contraction and relaxation. Then what we find is that the arrangement of the sarcoplasmic reticulum and the cell membrane is that, yes, the cell membrane develops and invaginates or goes inside in specific portions. This development of the membrane towards the center of the cell helps to ensure that the depolarization of the membrane of the cardiac cell or skeletal muscle quickly reaches the contractile units. Now, where it contacts the sarcoplasmic reticulum, it makes two structures, the tubule and the sarcoplasmic reticulum. So it makes two structures. So it is called a dyad. In the skeletal muscle, the T-tubule makes a triad because the membrane invaginates and develops into the muscle fiber where it contacts the sarcoplasmic reticulum and divides it into two sacs, one on either side. So the tubule in between and one sac of calcium on either side. Therefore, in the skeletal muscle, the T-tubule makes a triad. A triad meaning three structures. The tubule one and two cisterns or two sacs of calcium on either side. But for the cardiac muscle, it makes a dyad. It's just the sarcoplasmic reticulum, one sac, and the tubule. And also what we find is that the tubule diameter is 2 times larger than the skeletal muscle tubule diameter. And its function is to transport calcium from into the fibers, really. So this is just an illustration of the longitudinal section of the cardiac muscle. So we will have what you are seeing in dark spots, those are nuclei, and then we have those lines that are showing the junctions amongst the cells. So those will be called intercalated discs. So where the arrows are pointing, those are intercalated discs, which are the junctions where one cell, cardiac cell, comes into contact with the next cell and they communicate. And so what we see is that there is a central nucleus, often with a biconical clear area next to the nucleus. This is where the organelles and the glycogen granules are concentrated, and also this is where arterionatriuretic factor in arteriocardiac muscle is found. Again, you see that yes, there are striations or stripes because of repeated coordinated arrangement of sarcomeres. The cells are branched and separated by intercalated discs. Again, here we are just illustrating. If we did a cross section, we'll be able to see. If we did a longitudinal section, we'll be able to see. Again, here we are just trying to compare the skeletal muscle. If we did a section, transverse section or cross section of the skeletal muscle, we are seeing these are the muscle fibers with several nuclei. And then we have these as cardiac muscle on your left side of the screen, and you are seeing those separated by these connective tissue as well. So with skeletal muscle, it is delicate, highly vascularized connective tissue that covers each cell, what is called the endomysium. The endomysium in cardiac cell is, yes, they are also for each cell. The fibers then combine to make or are organized to make fascicles. These are covered by perimysium. Now, in cardiac tissue, the perimysium really is also there. And then the several fascicles are bundled into a muscle organ by epimysium. For the cardiac muscle, the epimysium is also referred to as the epicardium. Cardia related to, as in related to the heart. So this is just illustrating the tubule arrangement with sarcoplasmic reticulum. And this also is highlighting the fact that there are these coupling or interconnections or intercellular communications within cardiac tissue. And so what we have is that we have one cell and the other. You can see the striations, and then they will be connected one to the next in the intercalated disc. And so what are the features of intercalated discs? We have adhesion molecules where they come and they make sure that the cells stick together. So that those are desmosomes. Then we also have gap junctions, the spaces within which cytoplasmic material in terms of small molecules like electrolytes tends to diffuse from one cell to the next, and so they will allow exchange or diffusion depending on the level of depolarization or hyperpolarization of the different cells. So if one cell has influx of sodium, then the next cell has less sodium. So that sodium can easily flow into the next cell through the gap junction, and the next cell also becomes depolarized.